It's now my pleasure to introduce our speakers for the next sessions, continuing the spotlight on generic drug review challenges and solutions. So we'll be hearing presentations from Dr. Yang Liu, a senior staff fellow within the Division of Bioequivalence 3, Office of Bioequivalence in the Office of Generic Drugs. We'll also be hearing from Dr. Feng Wu, a senior pharmacologist in the Division of Quantitative Methods and Modeling within the Office of Research and Standards in the Office of Generic Drugs. We'll also hear from Liken Liang, or Dr. Liken Liang, a supervisory chemist within the Division of Product Quality Assessment 10 in the Office of Product Quality Assessment 2 within the Office of Pharmaceutical Quality. And we'll also hear a presentation from Dr. Yang Wang, a lead pharmacologist, acting deputy division director within the Division of Therapeutic Performance 1 in the Office of Research and Standards in the Office of Generic Drugs. So please join me in welcoming our next presenter, Dr. Yang Liu.
Thank you for the nice introduction, and good morning, everyone. It is my great pleasure to be here again, sharing some case study experience with all of you, both in person or online. So the story we want to share today, together with my dear colleague, Dr. Fang Wu, is the totality of evidence, including a PBPK modeling to support the BE assessment and the approval of Mesalamine DR tablets. So hopefully through today's presentation from both of us, we can deliver the following messages. Number one, we want to present this case study of an ANDA for Mesalamine DR tablets with some dissolution issue in some certain dissolution media. And also we try to share with you and help you understand OGD's BE evaluation based on the totality of evidence for this case study. And then Fang will share some detail about the PBPK modeling and how we are using it as a critical evidence to support the final approval of this ANDA. So here's my disclaimer. This presentation reflects the views of the presenter only and should not be should not be construed or represent FDA's view or policies. So first of all, let me just give a very quick introduction or a background refresh about the ROD product and what are the studies recommended by the PSG. So the ROD product is Takeda's Lealda DR tablets, 1.2 gram. It was approved in 2007 under the NDA number 22000. This product is indicated to treat colitis in both adults and pediatric patients. Now here's the PSG. I just took a quick screenshot of the study recommended by the PSG, which is published in June 2016. There are 3 pivotal studies recommended for developing a generic version of this drug. An in vivo BE study under fasting and under fat conditions respectively. And the 3rd study is an in vitro dissolution study. And I do want to highlight one thing is the design of the in vitro dissolution study is it actually includes 3 stages. The stage 1 and the stage 2 are called a pretreatment or prepared stages. And the evaluation stages are recommending using 4 different medias at pH 6.5, 6.8, 7.2, and 7.5 respectively. And in a later slide, I'm going to discuss why this is designed as such and what is the physiological reason. And now let's just dive into, because due to the time, we just dive directly into the case study. So in this ANDA, based on the data submitted, the reviewers found that the PK and statistical results for both the pivotal in vivo studies met the BE acceptance criteria. I do want to say one more thing about the in vivo study recommendation is not only the Cmax and AUC overall were evaluated. The PSG also recommend to evaluate the comparison between the test and and the reference drug for the AUC 8 to 48 hour, which is the partial AUC kind of representing the drug absorption in colon, where is the site of action. So that also brings up the question about the mechanism of this drug or the site of action. So this drug, Mesalamine DR tablets, is actually a local acting drug. Is used to treat ulceritis in the colon. However, the function is not done through like the plasma concentration. Is actually dependent on the local concentration of mesalamine delivered into the colon directly. So in this case study, the test formulation, it's going so fast, but anyways. The test formulation adopts similar mechanism of drug release. It's actually using the same pH-dependent release controlling excipient as the RLD. However, the dissolution profile, not all of them, but some of that actually are very, very different between the test and the reference product. I don't know, it's keep flashing. But we can see actually over here, I don't know. So, but this is just a quick review. The left two figures, the profiles, are actually the comparison for the lower pH value, 6.5 and 6.8. And the last one, which is very comparable, is for the 7.2 and 7.5. And in this table, we can also see the calculation for F2, or the dissolution similarity. We can clearly see that the dissolution are not that similar in the low pH conditions. And we're wondering why, and how big of an impact it actually caused in our evaluation for bioequivalence. So let's just go through why this actually is, the drug itself is, why is keep going? I didn't touch anything, but anyway. Okay, so this is the mechanism for this coating, because if the drug is not coated due to the quick dissolution and quick absorption of mesalamine, the majority of mesalamine will be absorbed before it actually reach the site of action, which is a colon. And on the right side, we can actually, I'm bringing the GI tract into three sections in terms of the fate of this drug. In the stomach and the duodenum, where the pH is relatively low, the coating will protect the mesalamine from early release. And in the middle section, which is the small intestine, the outer coating will be eroded and start to release the drug slowly, and it will still deliver most of the mesalamine APIs into the column to do its function. So in this slide actually is showing, the coding for this product. It's showing the function. It's actually telling the same story. Let's go back here. So it's the same thing. So the pH dependent or pH protection of the outer coating will make sure the drug stay intact in the stomach, and it will start to erode, as you can see on the left side. It starts to erode in the duodenum, and mostly will erode it and start to release the drug in small intestine. And per the RLD label, it also says the coating of the pH-dependent polymer film, which breaks down at or above pH 6.8, normally in the terminal ileum or terminal small intestine, and start to release the mesalamine. And how does it serve the function to make sure the drug mesalamine is actually released slowly throughout the whole large intestine or whole colon? It's actually based on another mechanism. But before that, here is another piece of evidence from the radioactive study showing where the drugs are actually released. As we can see in these figures, in the first figure is the T3, 3 hour, which is 3 hours after administration. We can see at the distal small intestine, we can already start to see some fuzziness around the tablet, which means the mesalamine, the mesalamine was labeled, the mesalamine drug started to release from the tablet, which means the outer coating is pretty much eroded and does not keep the tablet intact anymore. And as the drug goes into the small intestine, the large intestine, we can see the drug already start to go everywhere and start to do its function. And that's exactly what we are actually concerning about, is whether it is similar between the in vitro dissolution phases and the physiological conditions, whether it is representative. As we can see in the green bars, and we can see the drug actually go through the physiological conditions, it will be eroded. The outer coating, the red outer coating, will be eroded after it exits the small intestine. And when it goes into the distal column or into the column, the outer coating is pretty much gone. However, that is a little bit inconsistent with what we actually designed the in vitro study. Again, the in vitro study design at different pH levels is try to represent all the conditions that we may appear or the drug may experience in the GI tract as it delivers. And the difference here is for example, for the pH 6.8 condition, which is used as the evaluation stage, the drug actually didn't have a chance to be exposed to a higher pH value condition, just like in the small intestine in vivo. So our question is whether this is actually making a significant physiological impact. So the question is to correlate the physiological and PK and dissolution study as one big piece of evidence. What kind of tool can we use? And Fang will be discussing about that. But now I just want to give a very quick summary of what we have observed before we actually having the PK modeling kicking in. So what we have observed is the in vitro dissolution issue for pH lower than 6.9 conditions, and we are starting to see different dissolution profile between the test and the RLD product. And the in vivo drug dissolution released through the GI tract may vary due to the physiological conditions in different patients or healthy volunteers. And we actually recommend the applicant to conduct the the PK/PD modeling, IV/IVC correlation, and to use that as a supportive evidence to justify the lower pH value incomparable dissolution result. And that's where I'm going to stop and turn that into Fang. But before I do that, here is a quick question, is based on the data we have collected, either directly through the RLD labeling or through other literatures and evidence, is that true that the RLD product, the Leota DR tablet, will stay intact and prevent the drug release until it actually goes into column? Apparently, the answer is false. So this is not the case, especially due to the radioactive study. The drug already starts to release in small intestine. And here is, I just want to steal like a couple seconds from Fang. So here's my acknowledgement. I first want to thank Dr. Partha Roy, the OB director, who is very supportive about, this is a very interesting case, and he's, the leader for the office level discussion. I also want to thank DB3 management, April, Key, and Li Gong, who actually having a lot of great discussion with the assessment team and make final decisions. And OB management also provide very helpful insights and questions and comments through the whole story that I'm sharing today. Last but not least, we have the great supporting team from the DQMM, and I will let Fang to introduce more about the PK/PD modeling part. Thank you very much.
Thank you, Yang. Next, I would like to continue the second part of this presentation. I will introduce how the PBPK modeling can serve as a part of the totality of the evidence to support approval of the Mesalamine DR tablets. Here is my disclaimer. And learning objective, for my part, I will mainly explain how to develop PBPK model to predict a local drug amount by incorporating dissolution data at different pH. First, we want also to identify which dissolution is the biopredictive. A little bit of background on the GI locally acting products. As we know, assessing the BE for GI locally acting products is challenging because the systemic exposure may not reflect the drug concentration at the site of action. So the BE recommendations of oral locally acting drug products are based on the drug product properties and mechanism of action. PBPK models may be used to explore the correlation between dissolution in GI tract and the plasma concentration profiles and predict the local drug amount and support BE evaluation of such products. As Yang mentioned about these background of the Mesalamine DR tablets, it is indicated for colitis, And the product-specific guidance for this product recommends fasting and the fat PKBE study, and also the comparative dissolution studies at different pH. Of course, include a pretreatment stage 1 at pH 1.2 and pretreatment stage 2 at pH 6.4, and then evaluate the comparability at each different pH, 6.5, 6.8, 7.2, and 7.5. However, for this generic drug product, we found the F2 values less than 50 when comparing the dissolution between test and reference at pH 6.8 and 6.9. However, it's more than 50. It's comparable when the dissolution is over pH 7. But the test product and ROD were found to be bioequivalent for systemic PK under both fasting and fat conditions. Here comes the question, whether the slower release of test product at a certain pH would significantly impact the local drug exposure compared to ROD. And you have seen these PSG recommendation and know the conditions we recommended for conducting the in vitro comparative dissolution study. And in this, for this generic drug product, we found that at pH 6.5, 6.8, 6.9, the dissolution are not similar between test and reference, and the F2 is less than 50. You know that we use these in vitro dissolution testing over a range of pH to serve as a surrogate of in vivo drug release in the GI tract. And we see actually these tested product shows decreased release compared to ROD at pH less than 7, but it's comparable at pH over 7. Whether these will cause any significant impact on the BE for this local drug, GI locally acting products, we will use PBPK modeling to support providing some evidence. And we use the PBPK modeling to try to identify which dissolution condition is biopredictive. And we will also use PBPK modeling to predict the local drug amount in the colon and use population simulation to predict the percentage of drug absorbed in the colon. So the so here I would like to use a little bit of this flow chart to go over the how we develop, validate, and apply this PBPK model. We first use the intravenous PK data obtained from the literature to estimate disposition parameters. Then we develop the PBPK model for Mesalamine DR tablet. The intestine first pass effect was optimized. And then we also incorporate 3-stage dissolution data with pH 6.8, 6.97, and 7.2 as stage 3. Incorporate them into PBPK model and predict the PK, systemic PK. And then the PBPK model was validated using dissolution and the PK data obtained from literature or other ANDAs, and to validate this model. And then after validation, we use this PBPK model to see which one is which dissolution is biopredictive and predict the drug amount in the colon. These two figures shows that PBPK model can capture the PK well after IV bolus administration of 250 mg mesalamine. And it can capture the PK after oral administration of the delayed release tablet. And we use other data from literature, and we can also see the modeling predict well of the PK. And we, as shown on the right table, we use the data, dissolution data, and the PK data obtained from the other ANDAs, and we show that the PK prediction actually is pretty well, and with the prediction error less than 20%. This means that the model is validated using these additional data. Then we use the validated data to evaluate whether the dissolution data at a certain pH is biopredictive. As shown in these tables, we can see if we use the dissolution at pH 7.2 and 7.0 as stage 3 incorporated into PBPK model, then the prediction error is less than 22%, which means these conditions are biopredictive. But if we use the dissolution at pH 6.8 at stage 3 into the model, then we cannot predict PK well with the prediction error more than 30%. So we know that the dissolution at pH 7 and 7.2 at stage 3 are biopredictive. So then we incorporate this biopredicted dissolution into the model and predict the local drug amount in the colon and also the percentage drug absorbed in the colon. As you can see from the right top table, and the simulation suggests that the local amount in colon is similar between R and the test product, ROD and the test product. And the population simulations showed that percentage of drug absorbed in the colon is similar between the ROD and the test product, with 90% confidence interval of this TR ratio for the drug absorbed in the percentage of drug absorbed in the colon can fall within 80 to 125. This should demonstrate that there is a low risk of non-BE at this site of action for the generic drug product. These two figures you have seen previously with Dr. Yang Lu's presentation, and based on the formulation design, release mechanism, and GI physiology, shown here, we know actually the drug, the generic, both RD and the generic drug, they have the enteric coating. The coating will be break down after it enters in the small intestine, especially the terminal ileum, where the pH is 7. Then the core continues to pass into the GI tract and go to the colon and continues to release the drug. The colon, you know that the pH is around 6 to 6.7. And we can also see the drug, actually, in the scintigraphic figure, the drug release in the ascending column is at the ileum, terminal ileum. It's kind of consistent with what we are thinking about the release mechanism. These are also consistent with our PBPK modeling. And the PBPK modeling shows that the dissolution at pH over 7 is biopredictive, but not for the dissolution at pH 6.8, because the drug is already break down before it enters into the colon. So the pH 7 is more biopredictive, as demonstrated by PBPK modeling and also the release mechanism. So in summary, the PBPK modeling simulations supported that the 3-stage dissolution profiles at both pH 7, 7.2 at stage 3 may be biopredictive to the local and systemic exposure. The predicted amount of mechanism in colon and the percentage of drug absorbed in the colon, they are similar between ROD and the test product. So the totality of evidence based on the dissolution data, formulation differences, gut physiology considerations, along with the PBPK modeling results were used to conclude that this generic drug product has low risk of bioinequivalence at site of action compared to the ROD. So this work, as mentioned by Dr. Yang Lu, supports the approval of this mesalamine generic drug product. So here comes a challenge question. To evaluate the BE for locally acting products, the evidence could include the following: A, formulation and the dissolution comparison. B, gut physiology considerations and the release mechanism. C, PBPK modeling and simulations. D, all of the above. I will let you think about 1 minute. Yeah, 1 second. Then the answer is all of that, considering the time limitation. With that, I would like to acknowledge our colleagues in DQMM and also the reviewers from Office of Bioequivalence, especially Dr. Sharon Thomas, who did most of the modeling work, and the reviewers from OB, Dr. Yang Lu, Dr. Teresa Lee, and Gong, and they are valuable inputs and collaboration during the review of this generic drug product. Thank you. Then I will turn over to Dr. Li Kan Liang. Thank you so much.
Okay, good morning, good afternoon, and good evening. I'm Li Kanliang. So today I'd like to discuss the complexity of generic oligonucleotides, some key challenges in achieving and demonstrating sameness, and also impurity profiles and controls. And finally, I'd like to share with you some of the agency's efforts in addressing these challenges. Here's my disclaimer. The presentation reflects the views of the presenter and should not be construed to represent the FDA's views or policies. Oligonucleotides are short strings of nucleic acid with therapeutic effects. They can be fully synthetic or semi-synthetic, and they can be modified or unmodified. There are many ways to modify oligonucleotides, and modification could be at the 2' position or the 3' position of the ribose ring, or could be on the bases. And the oligonucleotide can be conjugated at the 5' end and the 2' position or the 3' end. The internucleotide linkage can also be modified, and in most cases, this modification is non-stereospecific, therefore leading to 2 to the nth power of diastereomers. This significantly increased the complexity of oligonucleotides. The ribose moiety can also be replaced with a morpholino ring. And the non-stereospecific modification by the PDA or the phosphorodiamidate in those linkages also leads to 2 to the nth power of diastereomers. I apologize for the busy slide here. This table summarizes the 20 oligonucleotides CDER has approved as of July 2024. The last column lists some of the complexity considerations based on the for generic oligonucleotides based on the RD labeling information. The PS and PDA modification of the linkages actually gives rise to from a few diastereomers to more than 1 billion diastereomers. The same regulatory requirements for generic small molecule drugs also apply to generic oligonucleotides, such as having the same clinical effect or safety profiles and identical active drug ingredients, which translates into active ingredient sameness for complex APIs such as oligonucleotides. Sequence chemical structures, diastereomeric composition, high order structures, and potency and activities, etc., are among the sameness considerations. For most synthetic oligonucleotides, sequences are well defined and controlled and can be verified. Regarding the structures, for the correctness and completeness of the modifications, including the position of the modifications, need to be demonstrated for generic oligonucleotides. The sameness evaluation also includes physical chemical characterization at the drug substance and drug product levels. Oligonucleotide impurities have some unique safety considerations, such as off-target effects, immunogenicity and inflammatory risks, etc. These risks need to be considered in impurity profile comparison and also impurity controls. It is recognized that binding of oligonucleotides to their targets are largely sequence specific. Then why is diastereomeric composition sameness required for generic oligonucleotides? This is because clinical evidence is not available to confidently negate the impact. And there are also some evidences indicating that stereochemistry and conformation of the oligonucleotides may impact their binding affinity and resistance to nucleus. For example, Iwamoto and others studied myeloimmersion and various stereochemically pure isomers and found that stereo configurations at the PS linkages substantially impact the pharmacological property of the oligonucleotides in their studies. Also, approved oligonucleotides counted all diastereomers as the part of the active ingredient. Therefore, unless adequate data is available to prove that it has no impact, diastereomeric composition sameness should be demonstrated for generic oligonucleotides. The graphs in this slide were adapted with permission from the ACS publication by Rasmus and others. The authors changed the RNS ratio at some selective internucleotide linkages of the 20-mer by using different activators. Several different methods were used to analyze the samples. Each curve on this slide represents more than 500,000 diastereomers. The first method did not give much information about the composition. Other methods gave, with wider peaks, with some distribution features. Let's look at the MICC method and compare the levels of profile changes from sample 1 to 2, and from sample 2 to 3. The levels of profile change look similar. However, from sample 1 to 2, the RS ratio was changed at only one additional chiral center. But if you look at samples from 2 to 3, the RS ratio was changed at 4 additional chiral centers. This comparison indicates that certain diastereomeric composition changes may not be proportionally reflected in the profile changes by some methods. To address the analytical method limitations, one may consider using multiple analytical methods with orthogonal separation principles that can provide detailed distribution profiles, combined with other orthogonal methods such as an NMR and with additional physicochemical characterizations such as the melting temperature or TM of an appropriate duplex from the oligonucleotide. The methods should be adequately validated for sensitivity, specificity, and resolving power. If the diastereomer number is low, one may develop methods to separate each diastereomer from other diastereomers and impurities. This way, one can compare the composition directly. To demonstrate sufficient sensitivity and specificity of the methods towards various diastereomeric composition changes, one may consider making multiple suitability test standards for method development and validation. These standards may have RNS ratios changed at a diverse set of a small number of chiral centers. If the methods are sensitive to such diverse and verifiable small changes in diastereomeric compositions, it is more likely that these methods may be able to detect the diastereomeric composition differences between the test product and the RLD. So for distribution profile comparison, one may consider comparing both overall and individual regions of the profiles. The acceptance criteria for sameness comparisons needs to be justified, ensuring comparable safety and efficacy profile as the RLD. The RNS ratio measurement at each of the chiral center after its creation is actually an indirect but effective approach to obtain stereochemistry information of your product, especially when the number of diastereomers is very high. And typical analytical methods may not be able to provide a precise picture of the diastereomeric composition. So it is important for firms to understand the baseline of their product for future potential changes and to ensure manufacturing consistency. This may be applicable to both the new and generic oligonucleotides. Typically, oligonucleotide impurities profiles are very complex with high number of impurities. Some impurities may be due to the incomplete modification or having extra nucleotides or missing nucleotides somewhere in the sequence. The complexity is further compounded by the high number of diastereomers in many cases. The large molecular weight of impurities makes it challenging to separate impurities with small structural differences. The resolving power of a single analytical method may be very limited, and co-erosion of impurities and isomers needs to be addressed. Oligonucleotides are excluded in ICH guidelines for impurities. The FDA has not published a guidance on impurity thresholds for oligonucleotides. And furthermore, the agency has not recommended structure-based classification of oligonucleotide impurities for safety evaluation and quality control purpose for generics. For impurity profile comparison and controls, multiple methods with orthogonal separation principles may be needed to address the co-erosion issues. The nature and location of the structural change in the impurities need to be determined. For example, for inotertion shown in this slide, an impurity with one methyl C unit missing could have 3 different sequences or structures. The exact location of the missing unit may need to be determined unless otherwise justified. If there's any ambiguity, one may want to see if it helps by conducting a comprehensive risk assessment and consider all the possible structures or impurities based on your process understanding. For impurity level controls, RLD comparison is the rule of thumb for generic oligonucleotides. Orthogonal and complementary analytical methods may be used. And when dealing with new impurities, higher impurity levels, proposed impurity thresholds, or impurity grouping approaches comprehensive risk assessment may be needed to evaluate their impact on safety and efficacy. Off-target effect, immunogenicity, and inflammatory risks, and general toxicity, and any other risks associated with the possible impurities may need to be considered. Summation-based grouping acceptance criteria without comparing and controlling the individual impurities levels may be hard to justify for generic oligonucleotides. Some examples including the retention time segment-based grouping, or N-1 or N+1 groupings, etc. Also, the fact that oligonucleotide impurities have higher molecular weights and therefore lower molar amounts than in the small molecule drug case may not be an adequate justification for requesting a higher weight percentage limit in the oligo cases. You may want to see if a comprehensive risk assessment based on the worst case scenarios may help justify some of the grouping approaches. For RLD comparative studies, 3 batches are needed at the drug substance and drug product levels. For evaluating the API from the RLD products, one may obtain the API by an appropriate sample processing method, such as extraction or dialysis, etc., depending on the need and the nature of the RLD comparative study. For diastereomeric composition sameness evaluation, sample processing should not change the diastereomeric composition of the sample itself. If a test is sensitive to formulation or process history, one may consider subjecting both the generic and the RLD samples to the same process. For intrinsic property characterization, formulation impact may need to be eliminated. For example, TM is an intrinsic property of a duplex, but can be impacted by the presence of ions, etc. Therefore, TM measurement may be done at the drug substance level. For siRNAs, TM measurement may be performed at both the drug substance and drug product level to understand the formulation impact and also demonstrate sameness. Sameness evaluation on 3 exhibit batches at release and shelf life, at end of shelf life may be necessary to support initial ANDA filing in a timely manner. One may consider evaluating a development batch for age considerations and submit the results as additional supporting data, provided that the development batch is equivalent to the exhibit batch in all aspects of CMC. CDER has various programs to facilitate and advance generic drug development, assessment, and approval. Some of the examples are listed here. The agency has published quite a few product-specific guidances on oligonucleotides to help generic product development. The agency also has internal and external research programs to advance the regulatory sciences. Here's an example of the research by our colleagues at the Office of Pharmaceutical Quality Research to address the challenges in analyzing co-eluting isobaric impurities. These impurities were difficult to differentiate and quantify by the conventional methods such as LC-MS. The OPQR work actually illustrated the possibility of quantifying these very challenging isobaric impurities. With that, I'd like to thank all the people and teams and officers listed on this slide. And here are some questions for you to consider. Question one, OM impurities have much higher molecular weight than impurities of small molecule drugs. And the molar amount of the OM impurities are lower, and therefore OM impurities should generally be given higher weight percentage limit than impurities of small molecules. True or false? So the answer is false. The next question, which of the following statement is true? Oligonucleotide binding is sequence specific. Stereochemistry of the OM modification need not to be considered in sameness evaluation. B, I can use 3 development batches of the generic OM for sameness evaluation and characterization for my submission, even if they are not true representation of the exhibit batches. And C, for complex sameness or comparative impurity profile evaluation, I can use multiple orthogonal analytical methods that gives quite different and detailed distribution curves or profiles on the same sample. Or D, currently I can justify my quality control of impurities in generic OM based on literature information about OM impurity classification without comparing them to the impurity of the ROD. So which one is correct? C, that's right. So to summarize, ROD comparison is the rule of thumb for generic ONs, and when necessary, use multiple orthogonal methods. Also, conduct comprehensive risk assessment to help justify proposed solutions. And as always, seek agencies' help via various channels. And thank you for your attention. Now I hand to Dr. Wang.
Good morning, everyone. So in the next 20 minutes, I will try to walk you through the journey of first approvals of several complex generic long-acting injectable products. Hopefully, I will tell a good story. So at the end, you'll be able to describe different types of long-acting injectable products. To recognize what are the regulatory and scientific challenges we have experienced, and some of them have been resolved, and we certainly recognize there are remaining hurdles, and have better understanding on what is the product-specific guidance and their role for supporting generic development. And lastly, but not the least, to be able to have a better sense of how the importance of GDUFA-funded research program and how much that really contributed to where we are today. So long-acting injectable products, those are the products that are designed to provide controlled and sustained release for days and months. So in this slide and next couple of slides, I'm sharing information data that published in a review paper I co-authored with my colleagues, Dr. Wenlei Zhang, sitting in the audience, and 2 industrial peers back in 2021. We aimed at giving a very comprehensive overview of the landscape of long-acting injectables, including new drugs and generics in 2021. So the figure I'm showing here to show you the LAIs and the therapeutic areas that have been used. And you can tell they are very, very beneficial in the therapeutic areas that would require chronic disease treatment or management, because they can significantly improve patient compliance. Compared to conventional dosage forms such as tablets or capsules, we have far less numbers of applications approvals, but then the complexity level and the diversity of dosage forms is also very impressive. So as shown here, I'm gonna walk you through a little bit like history of the LAIs here. So back prior to 882, the very first generation or generations of LAIs could trace back as oil solutions and then some micronized drug substance suspensions, such as penicillin G. So back then, it was already probably very challenging to bring those drug substance into a product that can be given to the patient. But I don't think back then, now it's 40 plus years ago, the scientists then had really clear concept of being long-acting drugs. I think the very first, by definition, long-acting with a very defined therapeutic dosing regimen should be the polylactide co-glycolide PLJ-based microsphere product, Lupron Depot, which was approved in 1989. So followed on that in terms of dosage form, in 2009, there was very first multivesicular liposome product. But that one was to deliver cytarabine, and then after that there was another MVL. But those two products, unfortunately, were very short-lived, not due to safety or efficacy, maybe more due to business reason. Then in 2009, going back to PLJ-based delivery system, the very first in situ forming gel or implant was brought to the market as Eligard to deliver the same peptide leuprolide acetate as the microsphere product in 2009, as shown on the top right panel. And that product is organic PLJ polymer solution, and the peptide is suspended in the organic given as an injectable suspension. Then upon administration, the dosage form would form a depot on the site. Then after that approval in 2011, the history always repeats itself, right? So it goes back to the drug substance suspension. And this time it was for paliperidone palmitate, which is antipsychotic, and it's the nano size instead of micro-sized suspension product. Follow that, MVL came back as what was mentioned in the commissioner's opening remark to market. This time is a very, very successful product for pain management, the bupivacaine multivesicular liposome. So in the 2021 paper, when we worked on it, we had a table to list all the high revenue LAI products. And back then, we still did not have any generic products approved. But that time we were already hopeful, because after that time was almost close to 10 years work, we already have in-house ANDAs that were under assessment. So we were very hopeful to see the first approval, which actually not one, we had three. We had three first generic approvals for LAI products for three separate ILDs. So those products were Sandostatin LAI, which is a peptide-containing microsphere product, and risperidone Costa, containing risperidone for antipsychotic treatment, and Vivitrol, which is an naltrexone microsphere product. So the Sandostatin LAI product is the product that containing a glucose PLJ polymer, which is very unique. Majority of the microsphere products on the market are using linear PLJ as shown here. So in the next couple of slides, I'm gonna share some of the regulatory challenges and scientific challenges we have had. So the regulatory challenge for PLJ-based LAI generics, because those are injectable products, parenteral. So per regulation, they need to be qualitatively Q1 and quantitatively Q2, the same as the reference listed drugs. However, for complex polymeric excipients in general, if you work with polymer, you know it's never a clean cut, right? The characteristic of the polymer is often a distribution, for instance, for the molecular weight. And the composition can also have certain variability. But then when it comes to PLJ, another layer of complexity is the products that are made by PLJ are really products by manufacturing. So depending on the manufacturing condition, methods, the raw material you start with may not represent the polymer you have in your finished product. And I'm having this figure here. It's in, I think, the Lupron Depot patent. by alchemists, showing you, depending on also your commercial scale, the level of degradation PLG can undergo is very different. So the regulation tells us, okay, you need to have the same inactive in the generic product, but it doesn't define what is considered as Q1 the same, and it's up to us to use the science to inform the decision making. And when we first started our research back in 2013, with the support of implementation of GDUFA, we knew nothing. That was the truth. So that regulatory challenge is translated into our scientific challenge. One, how do we define polymer sameness? What to do? PLG was not new, and PLG was very well known for formulation scientists. It started as a medical device for suture, and pioneered by, I think, Dr. the Lambert in MIT to be able to make high molecular PLG that is suitable for drug delivery. But most of the time, people do it to develop formulations by error and trial and error. So there is really no very systematically methods available to characterize the PLG and to inform. So we took the challenge and we funded our research program to look how we can reverse engineer the complex drug products and to identify the PLG and determine its characteristics. And we publish our results in a timely manner to facilitate generic development. Then I mentioned one of the first 3 approvals, the octreotide sandostatin LAR, used a glucose core branched PLG polymer. And there was 0 literature published back then discussing how we can potentially characterize this particular polymer and to prove if the glucose is really in the core. And when we first started working on this project with Dr. Ian Park at Purdue, they just used the conventional NMR, and we didn't see any signal. So we were like, what's going on? And is it really a glucose core branched polymer? And how do we characterize the branch frequency? All those things were very challenging. And honestly, when we first started, we didn't know if we'd be able to deliver, but we have to start and just keep going. But at the end, we were able to actually publish multiple papers providing characterization methods to characterize the branch structure of the polymer. And we also actually characterized all the commercially available raw materials to guide the generic development. Then, so this slide just summarizes what we have learned and our current standard to guide the Q1 polymer sameness assessment. So our expectation is we would like to see the polymers extracted from the finished products, both T and R, to compare their properties to ensure they have comparable characteristics. This is not a conventional approach or a routine ask for all polymeric excipients. This is mostly, like I mentioned, we want the applicant to really keep the endpoint at the starting line to realize your game is not simple. Sometimes we see data submitted on raw material, but if you are really having a raw material matching on face value, the properties in the finished ILD product, when your material goes through manufacturing, you may not end up with a product that has comparable performance. So it's really for the applicant's benefit to know this in advance. Then the Q1 sameness, at least for most of our products, we think we have done great progress and have resolved most of the difficulties, if not all. But then in recent years, we started seeing, because PLG polymers, they have well-established clinical safety profile. So whenever a company can leverage from this group of polymers, they tend to stay within this group of polymer without trying new excipients. But then it's not sometimes you use one kind of polymer could give you the desired release characteristics. So then there are plenty of smart people, right? So what they do is they start to mix different PLG polymers to fine-tune the degradation profiles to get what they want in terms of the release characteristics, which is brilliant. but then it's also going to be tremendously challenging for generic perspective. The first question we have is, if there's a mixture of two PLJ polymers, and how is it scientifically feasible for us to actually separate them? And if we can, to what extent? And what would be the most reasonable regulatory expectations in terms of the sameness in generics? So sometimes we do research, and that could, on face value, scare generic industry in terms of what are you trying to do. But sometimes we do things to the bottom because we want to know where should we draw the line. So we do something doesn't mean we're going to require the exact same thing as part of the generic development. But sometimes it's actually the opposite. If we have enough understanding, for instance, to show you don't really need to have the very detailed level of separation, and as long as you can meet the bulk property, that's sufficient, then that's our internal justification to develop the policy. So this is still ongoing, and we have made good progress, but we are not there yet in terms of clearly articulate where we stand when a mixture is used in a formulation. So next, the scientific challenge is the in vitro release testing. So this is a general challenge for long-acting products because the IVRT itself just very long, and because the release mechanisms also can be very complicated. So sometimes depending on the method condition, for the same formulation, you can get very, very different profiles. I think yesterday there was a good discussion in the IVRT session, right, talking about then what should we aim for? Should we aim for like a profile condition that mimic the in vivo PK, or should we have a condition that we know that is more discriminatory for differences in formulation characteristics, composition, or manufacturing parameters? I think those are all very good and valid questions, and that's part of our research to really first to explore all the scientifically sound methods, what are out there, and then what may be more suitable for QC versus for BE purpose. So here for long-acting injectable, we have been working on, for instance, looking into real time and then also accelerated time, because we understand how time is valuable for drug development. So we looked into develop important tools to understand the impact, how the formulation parameters, like the source of the polymer, if you purchase from supplier A versus B, if they give you the same performance, and then also use that to explore novel B approach. For PLJ-based microsphere products, we are not there yet, but we have ongoing research for instance, in situ forming, which we are trying to see if we have enough understanding to push for totality of evidence based in vitro only approach for supporting BE, or as a in between, if we can have in vitro in combination with in vivo, then reduce some level of burden at the clinical site. And ideally, once we have methods, IVRT methods that we're really comfortable, we will start to maybe implement them into our PSG to be more descriptive so company can follow. And we also start to include references in our PSG so company can directly look at the papers we have published. So challenge 3, bioequivalence study, is already sort of part of the IVRT. In general, the BE study, our expectation it should be accurate, sensitive, and reproducible approach. So for PLJ-based long-acting drugs, so far, most of the PSGs we recommend in vivo BE studies with PK endpoint. We have one PSG for dental product that is a locally acting product to recommend comparative clinical endpoint study. And we actually understand and know the challenges associated with that approach. But that was the most viable approach at the time of PSG development. And I think that goes back to another question raised in yesterday's session. why FDA cannot just recommend a PSG with the most sound approach instead of keep revising it. But I think the fact is, when something is complicated, it's complicated for everybody, for industry and for FDA. So it does take everyone's time to really understand and come up with an approach and keep improving upon it. And that's what we have been doing. So for the dental powder product, we have been using that as a model product to study, and we're really getting close to include an alternative in vitro approach. So this slide here just give you an example PSG. This is with partial AOC because for long-acting products, we often also recommend partial AOC instead of just conventional PK, excuse me, to make sure the PK profiles are comparable, but also the sustainability of a product is also similar. Sorry, excuse me. Then the next scientific challenge or efforts, I would say, we have been working on is the IVIVC. So here I mentioned a couple of times already, totality of evidence in vitro approach. So IVRT is always part of the approach, but because that is a totality of evidence approach, so as far till today, the PSGs for complex LAI products, in which case we recommend in vitro only approach, the IVRT was not targeting IVIVC. So IVIVC is always desired, and it in theory can be a standalone study. But as totality of evidence approach, the IVRT is more targeting to demonstrate understanding of the drug release mechanism instead of really strictly targeting IVIVC conventionally to predict the PK profile. So there is a subtle difference. I'm not discouraging IVIVC, and it can be very beneficial, but just want to point out our totality of evidence, the IVRT, is not necessarily say you have to have an IVIVC. But in order to develop IVIVC, so the paper I'm showing here was published by Dr. Diane Burgess in UConn, and they have lots of experiences exploring IVIVC for complex LAIs. And in recent years, we also start to collaborate and exploring advanced analytical technology to better understand the microstructure of those polymeric systems, so that can inform really the release mechanism. So I'm showing you a few figures here. So we use, for instance, the iron beam focused SEM, and then we take the image and use artificial intelligence informed data segregation to reconstruct the inner structure, and then couple that with our IVRT data to really understand the drug release mechanism. So this slide here, I just give you a quick walkthrough in terms of the timeline of our GDUFA research efforts and how OGD FDA progressed to where we are for the first generic approval. So back in 2013, with the implementation of GDUFA 1, we initiated our first batch of research activities focusing on PLGA-based LAI products. Then in 2015, the first paper published describing analytical methods that can be used to reverse engineering the PLGA-based RLDs. And then in the same year, we revised previously published PSG for risperidone Costa to include specific recommendation on IVRT and further updated our recommendation for the PK study portion. And then the first guidance for octreotide LAR, naltrexone, and risperidone was published or further revised in third. These. Besides the ENDA assessment and our PSGs, then the research outcomes has also been used to address Yeah, I guess the milestone slide wants to have a little bit more highlights. Okay, so the next couple of slides, really I just quickly mention the MVL, which you have heard multiple times. So this is the picture again, and this is a mixture of liposome and microspheres. So the particles themselves are more like microparticles, and they are really in the micron range, and they are sphericals. But then within the spherical, you have this honeycomb structure where the lipids are forming small compartments or chambers. And then that particular packaging has the conventional liposomal feature. So the complexity of this PBPK MVL is a lipid-based microparticle with nano-sized inner structure. It's very complex in terms of manufacturing. I will show you, we started our internal research, I think back in maybe 2015. And the FDA lab, we found maybe close or more than 30 ways to fail the manufacturing. So that's how many times we have tried and failed. And it's really a big shout out to the lab fellows. Just don't give up until we had a very decent formulation that has this honeycomb structure with reasonable stability. And then this is a locally acting product. Systemic PK is also surgical site dependent. Yeah, then in 2017, we published a paper presenting a very novel IVRT method. Until today, we haven't seen a replicate. What's unique about this method, which made the cover story of Journal of Control Release back then, is it's a testing method coupled with inline UV measurement to continuously monitor the drug release. And we were able to find a condition that really reflects the multiphasic release profile of this MVL product. Its PK profile, as you can read in the labeling, is multi. It has two peaks. So it does have a lag phase before the secondary peak or drug is released. Then based on our research, we published our PSG in 2018, and then the first generic was approved earlier this year. But then you can tell from 2018, we didn't really stop there. We know what we recommended in our PSG, and back then we didn't really have all the data to prove the feasibility, or also we want to help generic to better understand our methods. So we had external research carried out reverse engineering and characterization, and we published a paper sharing all those analytical methods. So in today's presentation at the end, I don't have an acknowledgement slide because it's really just not possible for me to have one slide listing all the names, all the individuals that have contributed to the LAI research area and internally and externally. So as a tribute, I'm having two slides listing over 40 publications that were produced through the GDUFA research by our internal external collaborators, and also can be a good source for the audience if you are interested in reading some of the papers and learning more about what we have learned. And for my challenge question, it's very simple. Just want to see if everyone thinks all long-acting drug products are considered complex products. True or false? Yeah, it's false because, like I mentioned, the very first generations, the oil solution, those are not complex. Then for drug substance suspensions, they are complex products and they are very challenging to develop as new drugs. But within that, depending on the clinical risk, penicillin G versus paliperidone, we will not consider them at the same complexity level. So what I want to say here is really, when we think something is complex or not, you really have to take this holistic, 360 angle to look at all the aspects rather than fragment and just look at one aspect. So with that, I will summarize. Polymer or lipid-based microparticles, those are very complex, long-acting injectable products. GDUFA research projects have made great contribution to improve the understanding of these products. Those outcomes we have used to develop our product-specific guidances and to address the regulatory inquiries to support the generic development and approval. And OGD will continue to improve PSGs based on updated understanding obtained through communications with generic industry, workshops, CCs, pre-and meeting requests, and GDUFA-funded research. So I think this is really a collaborative effort. We take the first step to publish a PSG to reflect what we think at the moment, maybe the best approach. But that doesn't mean that is truly the best per your perspective. So we really need to hear industry's feedback, criticism, challenges. Those are fine because those are the things push us forward and we should work together and communicate more. So that's why we highly encourage generic industry to really leverage our GDUFA research outcomes and engage with us early during development. So with that, I would like to thank you for your attention and invite all the speakers to the podium. Thank you.
All right, thank you again to our presenters for their wonderful presentations. We'll go ahead and start our question and answer panel to include all our presenters from earlier, and also joining the panel, we have Dr. Hansung Chen, a senior interdisciplinary scientist from the Division of Product Quality Assessment 12, within the Office of Product Quality Assessment 2 from the Office of Pharmaceutical Quality. So thank you again for joining us. As before, attendees in the room are invited to line up at the mics to ask your questions. And then for those online logged in, if you haven't had a chance to submit your question already using that on-screen link and QR code, please do so now. And we'll answer as many questions as we can. So first, we'll see if anyone in the room, again, wants you to come up to the mic. If not, in the meantime, we'll go to our first question online.
Our first question is for Dr. Yang Liu or Dr. Hansung Chen. How was IV-IVC for mesalamine able to capture variability? How many different rates of release formulation were assessed? And can you provide more information on unit impulse response formulation used for deconvolution?
Okay, so can you repeat the second part of the question, please?
Sure, yes. The second part was how many different rates of release formulation were assessed? And then can you provide more information on unit impulse response formulation used for deconvolution?
Yeah, I think maybe Fang is the expert for IVFC, so I'll let her just chime in first.
I think the first question is about the variabilities and also how many immediate delayed release formulations assessed. I think for this one, we focus on the mesalamine delayed release tablets. The ROD is Liada. So we focus on these ROD and the relevant generic drug formulations. We use these as a case example for indicating how PBPK can be used to identify the biopredictive dissolution and predict local drug amount of drug in the colon. So yeah, and regarding the unit pulse for the disposition part, I think they are asking about the disposition. Actually, we use the two-compartment model, and to get the disposition on the PK parameters. The two compartments we have K1 to K21, and also the clearance that we put into the model, and to estimate disposition parameters. I hope that answered the question.
Yeah, thank you, Fang. I'll also chime in a little bit more from the formulation perspective. So for this drug, Mesalamine DR tablets, referencing the same RLD product, there have been several endo versions or generic version approved by the FDA. From a formulation perspective, these formulations include the one we have in our case study. They all use very similar excipient, the pH-dependent excipient per se. However, these approved endo products, for the pH-dependent excipient they use, they have different amount or they have different ratio. So if we're looking back into the in-house data we have been reviewing for this generic product, we are observing some very different dissolution profiles, either between the RLD and the test or across different endos. So that also reflects the variability of this kind of a complex mechanism of release. However, as Fang just mentioned, these data or this variability or the slight different design of the formulation were taken into consideration when we put the in vitro dissolution data into the PBPK modeling to get a supportive evidence as one of the piece of evidence we use to support either the approval or the decline of the endo.
Thank you so much for all that information provided. We'll go to another online question.
All right, this question is directed towards Dr. Lichen Liang. For products like Defibrotide, which is a polydispersed mixture, and RLD is expected to have a lot of variability, how is sameness comparison performed if the RLD itself is changing?
Thank you for the extremely good question. Actually, I didn't have time to talk about Defibrotide. Defibrotide is a little bit different from the other oligonucleotides because the RLD sequence itself is not defined because it's a mixture of different combinations of sequences. This is by control depolymerization of the DNA from the porcine. So itself has a combination of all different sequences. So instead of trying to compare the sequences, which is very challenging, actually we published guidance and actually advising the industry to actually attack this from different angle. If you use the same source of the the animal tissues or organs and things like that, from the same source, and also use a similar manufacturing process, the same mode of depolymerization, you may have a better chance to have similar kind of distribution on the sequences. So this is quite different from other oligonucleotides. Yeah, thank you. I would highly recommend that you check the PSG for advices. Thanks.
Thank you again for that response. A reminder again to people in the room here, remember, this is your opportunity to get your questions answered right here, right now. So feel free to come up again to the mics. If we don't have any takers for the moment, we'll continue with our online questions, our next one.
All right, we have one for Dr. Yan Wang. Is it acceptable to establish and submit IV/IVC analyses using animal models for LAI products due to the long half-lives for LAIs?
Yeah, thank you for that question. I guess the key in that question is the animal data. So I think as of now, animal data, how we view for generic development, we are certainly open, to get that piece of evidence as like supportive or applicant could use that. And that's what we have been doing in our own research to obtain a baseline understanding of what may be the drug release mechanism and behavior, although species translation is always technically challenging. But as of now, if it's intended for regulatory purposes, I don't think animal data is accepted to support generic assessment or approval. But for development purpose, we are open to look at the data, for instance, in pre-ANDA meeting mechanism and provide feedback to help maybe better design the PK or whatever is relevant to the development.
Thank you again for that response. We'll go to another online question.
Of course, this one is directed towards Dr. Hansong Chen. Is it acceptable to develop a dissolution or IVRT method different from the USP monograph ones?
Thank you for the question. Yeah, this is very general question. The short answer is yes. Yeah, you can develop a different method from the ones listed in the USP monograph. I would like to add a little bit more about this. Our current thinking is, the dissolution method or in vitro drug release method is product specific. So, when you develop the dissolution method, the USP method or FDA database method, it's just the starting point. So you should develop your own method that's appropriate for your drug product. So our expectation is, the method should be sufficiently discriminating against the CMS and the CPPs. So ideally, the method should be clinically relevant. Yeah, thank you.
All right, thank you for that information. Again, still open for anyone in the room to come up if you'd like. But if not, in the meantime, we'll still go to another online question.
All right, we have one for Dr. Fong Wu. For mesalamine, if PBPK modeling shows that dissolution in media less than pH 7 is not as relevant, can the recommendations be changed to remove dissolution testing in these media?
Thank you for the question. So yeah, as we showed that for the Leada and this specific drug product, the dissolution at pH less than 7 appears not be predictive. However, that this dissolution data submitted to the agency still can be considered as some supporting evidence, or we can also consider look at the data and to see whether there's any difference, and also we consider the release mechanism about these drug product. And then for this product we use the biopredictive solution mainly to put into the PBPK and predict the local drug amount. So we still need that the data at less than 7 for looking at the data. That would I comment. Thank you.
If I may, I also want to provide a little bit comments for that question. So so as I like what Dr. Wang and also Dr. Wu was talking about, and the PSG actually is reflecting the current thinking or the growing understanding on certain aspects based on the knowledge and experience we collected in our review, in our internal research job. Regarding the mesalamine DR tablets, dissolution data under conditions that is a little bit more acidic. So my comment is, so the recommended evaluation stages in the PSG with the 4 different pH values is try to represent all the possible conditions after the drug actually was delivered into the small intestine and after. So this is, you can say this is a more conservative approach. If a test product can have a very similar dissolution in all these conditions, that would be the ideal case. However, just like Fang mentioned, if the applicant observes some issues or some incomparability between the test and the reference in, for example, 6.8 pH condition, it's still recommended to submit the data, and the agency in our review or in the follow-up meeting with the applicant, we are open, just like the case study we shared today, we are open to consider and take into account other evidence, including but not limited to PBPK modeling, to support and to evaluate the bioequivalence from a totality of evidence perspective. So hopefully that is clear. Thank you.
Thank you so much for that response. And it looks like we have a question in the room. Go ahead.
Yes, thank you, Marie Ducharme, Learn and Confirm. It's a follow-up to the previous question that was online. It seemed to me that it made sense that you've proven that 6.8 was not predictive, that you would remove it from the individual product guidance. It seems to me that made sense. Going back to your answer, where you said that the 6.8 was not predictive, here is a question for you. In your PBPK analysis, if I understand correctly, you kind of said that the predictions were validated because you were plus or minus 20% on the mean. And I think I heard that you decided that it was not predictive because when you included the 6.8, you had plus or minus 30%. Okay. My question is, this product is highly variable, right? Correct? It has 70% to 100% intra-individual variability. Correct? So why would plus or minus 30% not be good when it's highly variable at plus or minus 70 to 100%?
Yeah, so first I would like to clarify, and just now we said that although we showed that the solution at pH 6.8 is not biopredictive, but this is still included in the current PSG. And also we are currently thinking is that data can still be submitted to the agency, and we are going to look at it for evidence and considering the study, the formulation design, release mechanism.
I understood that.
Yeah,
I understood
so
that.
just not to be removed from the PSG, just to clarify that. And then for the variability you mentioned, yes, the Mesalamine, it is a highly variable drug product. And what we generally consider, which is biopredictive, is based on our current practice on using PBPK modeling, especially for the BE assessment. If the prediction error is more than 20 or 25%, that considered is not a good prediction. So that based on that, we considered that the pH 6.8 included that is not biopredictive as compared to the observed PK. Yeah, and yeah, I understand.
I'm sorry, but in previous presentation by the agency, validation of PBPK is usually plus or minus twofold.
So yeah, I think
I'm really surprised that you're accepting plus or minus 20% and then rejecting plus or minus 30% when this is a product that's highly variable. I mean, it's a little bit surprising.
Yeah, it's not rejecting, just comparing to the other dissolution that they are.
But that's how you decided that 6.8% was not, that 6.8 was not relevant. That's how you decided that, because you got plus or minus 30%. I mean, think about it. I just, anyway, a comment.
Yeah.
For thinking.
Yeah, thank you. Thank you for the comment. Yeah, for the twofold, actually, we are considering whether it is validated or not, depending on the modeling purpose. The twofold, yeah, as you know that the DDI, when we use the modeling to see whether it is validated or not, sometimes twofold we can consider, and also if there is a the platform validation, we can consider that big variability, the prediction error. But for the PE assessment, generally we consider like 20%, around 20%. Yeah, thank you.
Thank you for that. We have another question here. Yep.
So I have a question for the PLG, the long-acting drug. So the PLGA. The PLGA is an excipient, and there's a lot of study, a lot of focus from a talk. So from a previous talks, especially yesterday, it's API has been focused a lot. Your talk, PLGA, has a lot of research started there. So my question is, what reason behind that? Just you want to make sure the release duration is okay, or you also talk about impurity or something else? Why that has so much focus?
So your question is why we spend so much efforts on characterizing the PLG, considering it's being
Just
an
excipient,
excipient?
yeah.
Okay. Yeah, I think that's a fair question. So first of all, PLG is the excipient and it's actually a functional excipient, meaning its characteristics plays a very important role for the drug product performance. Then the main reason for us to spend so much efforts on understanding the polymer, one part is because its impact on the product performance, then that directly relates to the successful rate of having a successful product. But the other hand, as I mentioned, is to fulfill our regulation. So the regulation, because the PLG-based products, most of them are parenteral products. So the regulation requires the generic to demonstrate the composition of the generic is qualitatively and quantitatively the same. But then for PLG polymer, because of the inherent variability, is not as straightforward as other, some excipients Q1, it strictly goes to chemical identity. So our efforts is really to understand the characteristics of the polymer and the source impact of the polymer on the product performance, and also to guide. Right now, we have made great progress in terms of we have clear guidance to tell the applicant what they should look for, what they should do. But then having said that, we also understand from generic development standpoint, it's still very restrictive in terms of today, probably in terms of characteristics, the molecular weight, we have probably the max flexibility depending on the product and our understanding. So if we know a product, the PK and its release is not so sensitive to differences in molecular weight, mean molecular weight and distribution, then on face value, if we see a 10 to even 20 KD difference in the mean molecular weight, we would still consider that as the same polymer. Then, but in some other cases, depending on the product or dosage form, we also know if you have less than 5 KD difference, you will see a difference in the release and the PK. Those things may not be obvious to applicant depending on how much experience they really have. And that's why we do those research and publish, share our understanding, and also we provide as much detail recommendations in our responses to their inquiries to facilitate their development, to improve the successful rate, because those are so complex and costly to develop, they probably don't have many chances for failure.
Thank you. I guess we can talk a lot on that in offline.
Yeah, sure.
All right, thank you again for that response. Maybe we'll try to fit in one more online question.
Sure. So we have a question for Dr. Yang Wang. So are there any validated mathematical models for establishing IV-IVC for long-acting injectables?
A very good and challenging question. As of now, I think the IV-IVC we have successfully demonstrated in animal, those are more empirical mathematical model. So either time shifting or time factoring based on mostly, the release mechanism. If it's sigmoidal triphasic release, we would try to keep the shape of the in vitro, then just compare the time scale and factor that. Having said that, there are ongoing efforts actually in the sister division, DKMM, to look into PBPK, for instance, those model to establish IV-IVC mechanistic modeling. And also from our perspective, like I mentioned briefly, we are also looking to like the physics, the AI-based simulation, and then the imaging microstructure supported release mechanism exploration. And we are looking to using those tools to establish IV-IVC. But at the moment, I don't think we have a robust, validated mathematical model that can fit all products.
All right, thank you again for that response. And I know we're going a bit into lunch break, so maybe we'll go ahead and end our session there. But thank you again to our presenters and panelists for all the excellent information and insights. Let's go ahead and give them another hand. Yep.